Okay, so we'll get started. This afternoon, it is our pleasure to have a speaker from, well, in-state, let's put it that way. You'll hear the details there. And as we normally do, we have one of our students introduce our speaker. And today, the introducer is Miss Joya Dean. And she is biochemistry on the pre-med track. This is her second year at Andrews, third overall. Her hometown is Lansing, Michigan, and she went to Lansing Christian School. She plans to go into medical school, following in the footsteps of several of her family. Are you saying like close family, mom, dad, uncles, aunts? Is that what you're talking about there, Joya?
It's most of my aunts, and then I have a bunch of extended family, like great uncles and aunts who are all like in the medical field.
In medical field, okay, very nice. And so, Joya, in terms of fun things, what do you do?
If I ever have any free time, I'll probably just like lay in bed or like watch something on Netflix or YouTube, just like lie down.
Yeah, these chemistry teachers, man, they kind of take away your fun time, right?
Just a little bit.
Just a little bit. Okay. Well, it's your turn to introduce our speaker for today.
Okay, thank you for that introduction. I'm introducing Dr. Brittany Morgan. She earned her Bachelor of Science degree in biochemistry from Western Kentucky University and earned her PhD in chemistry at Duke University. During her time at Duke, she worked with RNA properties to design RNA-targeted libraries. Dr. Brittany Morgan has received many awards from her training, including many fellowships such as the Barry Goldwater Scholarship, NIH T32 and F32, Michigan Life Sciences Fellowship, and most recently, the Burroughs Wellcome Career Award at the Scientific Interface. In addition to these accomplishments, Dr. Brittany has been recognized for her dedication to mentoring, outreach, and service, giving her the Dean's Award for Excellence in Mentoring. Dr. Brittany Morgan is currently a Michigan May Walt Life Sciences and NIH Ruth L. Kirkstein postdoctoral fellow in the Life Sciences Institute at University of Michigan. In July, she will begin her independent career at the University of Notre Dame as the John V. O'Connor Assistant Professor of Cancer Drug Discovery. Her lab will be dedicated to targeting dynamic and disordered structures in RNA-binding proteins that are aberrant in cancer. Today, Dr. Brittany Morgan will be talking about therapeutically targeting dynamic proteins. Please join me in welcoming Dr. Brittany Morgan.
All right, very good.
Thank you, Joya, for that kind introduction. And I'm excited to see your journey in medical school. Sounds like you've got a really great support system with your family too, which is awesome. Yeah, so I'm excited to tell you all today about therapeutically targeting dynamic proteins. But I want to start out with a couple of questions for you. So how many proteins do we have in our body? Does anybody know the answer to that? Just take a guess. You can put it in the chat or feel free to unmute yourself as well.
I just learned that we have less than 25,000 genes, but I can't remember how many proteins.
You're pretty close.
Oh, okay.
Yeah. We have right over 20,000 different proteins within our bodies. So thinking about drug discovery, how many of these proteins do we think we've successfully targeted with drugs? Think about a percentage. What percentage do we think we've targeted with drugs?
Low.
How low are we going?
Maybe less than 10.
So we have 40% in the chat.
40% in the chat.
So we've actually successfully drugged
Whoa.
4%
4%.
of those 20,000 proteins. So it's a very, very small percentage.
Very small.
We have two more questions. So how many of these proteins have we successfully made ligands for? When I use the term ligand, I mean a small molecule that targets the protein. Maybe it's just in a test tube, maybe it's in a cell, maybe it's in a mouse model or in clinical trials. It's just not an FDA approved drug just yet.
Oh, okay.
What percentage do we think of these 20,000?
Oh. Is it like a percentage of the drug? No? Oh, okay.
No.
Just, okay.
Total.
I don't know.
We have a 5% in the chat.
Yeah, yeah.
Let's take, so it's actually 11%.
Okay.
Of all of these proteins we've successfully made a small molecule for. And there have been a lot of scientists that have asked, well, if we use all of our toolbox, all the tools in our toolbox, how many of these proteins can we target with small molecules or small molecule ligands? And those estimates are just in total 20 to 40%. So that means that there's an entire proteome that we currently can access using all of our tools that we have. So of those 20 to 40% of the proteins that are what we call ligandable or druggable, many of these proteins have pockets for the small molecules. We often use the term lock and key. You think about, we have a lock on our front door and we have a key that has perfect shape complementarity to unlock our lock at our front door or to bind to the protein of interest. So for these other 60 to 80% of proteins that we can't target right now, most of those lack those traditional pockets that we think about. And many of them are dynamic or disordered. And really the question is, well, how do we make keys? How do we make small molecules for things that don't have pockets and that are constantly moving and don't have structured structure? And that's really what I'm interested in, what I've been interested in for my entire scientific career. So I want to start out with an example of how challenging these 60 to 80% of proteins are to target. You may have heard of the family of RAS proteins. This is one of the deadliest families of cancer-causing proteins. You can think, so looking at just this graph, this is the percentage of various cancers that have mutations in the RAS protein. And these are some of the deadliest, like pancreatic, colorectal, lung cancer. And people have been researching how to target RAS with small molecules for close to 30 years. And we've literally spent billions in funding. And we have no FDA approved drugs to target this really important class of proteins. And this is one example of this protein. And there are what we can see potentially like a pocket located here that maybe a small molecule could bind. But the challenge is that these pockets, where they form, the structure is really dynamic. So we can think about it as a lock on our front door that's constantly changing its shape. How can we make a key for that? So there was a success actually last year, having the first FDA approved drug for RAS protein. It was for KRAS that had this glycine to cysteine mutation. And so I have the structure of glycine as well as the structure of cysteine. I only have two more questions for you all today, and this is one of them. So when it has this glycine to cysteine mutation in the protein, what property can you all think of that we introduced with this mutation?
Cross-linking?
Yes. So we introduced a great nucleophile
Yep.
into our protein. So we have an example of this small molecule that binds in this really nice pocket. And because of this presence of the cysteine, we can then form a covalent bond to the protein. And that we can think about it as the lock that is constantly moving, and all of a sudden your key is permanently attached. And that little key was really important for leading to having the first FDA approved drug for this really horrible class of proteins. It was approved in 2021. And this initial discovery actually has led to 18 other small molecules that are in preclinical or clinical trials for RAS proteins. And it was really that covalent bond that was super important for the discovery. So going back to this idea of the 60 to 80% of the proteins, I showed one example today and how much of an effort it took to have a drug for this protein. And really thinking about what we learned, that these pockets that do form in these dynamic and disordered proteins, they can be transient. They're not there all the time. They can appear sometimes when the structure is moving, sometimes it's not there. And also sometimes these pockets, we call them cryptic, or maybe they only show up when the ligand is bound, so we wouldn't see them in the unbound protein form. So using these ideas, I've been studying a different protein, a transcriptional protein in my postdoc called MED25, and understanding, can we use covalent ligands to target it? So this transcriptional protein, I just want to take you back to the central dogma. For those who may not be familiar with biochem, we have DNA that's transcribed to RNA that is translated to proteins. The majority of the drugs that we have today often target the protein and alter its function, and that leads to a phenotype or a curing of a disease. What my lab that I work in currently does is we actually try to target transcription. And when we do this, we change the amount of the protein that's actually made in the cell. It's a different way of thinking about drug discovery. But one of the challenging parts of doing that is that all of the proteins that are involved in transcription are dynamic and disordered. They're all in the 60 to 80% that we currently can't target. And this is just a schematic of what that process looks like. So we have our DNA. We often have first a transcription factor that binds, and then the remainder of the machinery gets recruited to turn on gene expression. We then get the RNA that's then translated into the protein. So the idea is potentially, say we have a tumor suppressor and we want to turn it on in a cancer cell. Well, maybe we can alter these proteins in a way where we turn on its transcription. Or it could be the opposite. Maybe we have something like an oncoprotein like RAS, where we want to turn off its transcription so it can't lead to cancer metastasis. So with the transcriptional protein that I work with, it's called MED25. And it forms a lot of protein-protein interactions. And what's most important for those interactions are these really large loop regions that are found in the protein. And it's actually these loops that control what protein-protein interactions are formed and what genes are transcribed. And the genes that are transcribed are really important for a variety of things, particularly in viral replication, which we know from the pandemic that we're currently still in. We need better antiviral drugs. And also the proteins that are transcribed are also really important in cancer metastasis, which is another really critical need that we have in drug discovery. So can we target these loops in MED25 using covalent ligands, potentially to stop the transcription of oncoproteins or proteins that are important in viral replication? So similar to KRAS, I would say even more so, this protein is very dynamic. It has a lot of movement. We have a cysteine residue as well that's located, luckily, in one of these really dynamic loops that we can target. And I want to go back to that idea of why is that cysteine important? Why is the covalent bond important? So it allows us to increase the potency of the small molecule. So you can envision if the covalent bond wasn't there, you would have your protein, your small molecule, and it would continually be dissociating because the structure is changing. We don't have that perfect lock and key. However, when you have the covalent bond that forms, the only way that the ligand dissociates from the protein is when the protein is actually degraded. So it's always there. It's a very potent ligand. And also, it allows us to bind or to focus on particular structures. So the only cysteine present in this protein is actually in this dynamic loop. So then we can know what regions we're targeting. And we know this loop is important for function. So we have both a cysteine as well as a location that's important. So we performed a covalent screen to see if we could find molecules that form covalent bonds with the cysteine. So the technique that we used is called tethering. So tethering is a reversible covalent technique where we have the thiol from the cysteine located on the protein surface. This then disulfide exchanges with small molecules that have disulfides, where the ligands that have the greatest affinity for the protein are enriched in the solution. And we do this in the presence of beta-mercaptoethanol. Some of you all may have used this in the lab before. It smells really, really bad. And this is a reducing agent that allows this reaction to be catalyzed. So it's a covalent reversible technique. And one of the things that's really cool about this is because there's a covalent bond that forms, the mass of the protein ligand complex is different from the mass of just the protein alone. So we can use mass spectrometry to screen these small molecules. And we can calculate the amount of protein that's bound to the ligand by using the abundance by mass spectrometry. So we simply take the amount or the abundance of Med25 that's bound to the ligand, and we divide it by unbound Med25 and Med25 bound to the ligand, multiply that by 100, and that's our percent binding. It's a really easy screening technique for a lot of these challenging proteins. And we assume the higher the tethering percentage, the greater the affinity for the protein. So we performed this screen with 1,600 different small molecules, and we were able to find 12 leads that bound to this cysteine 506. And one of the first things that I was interested in was, okay, do these leads have the features that are common to the lock and key model that we often think about for our ligandable or targetable proteins? And these are features like shape complementarity, as well as things like electrostatics, like positive and negative charge coming together to form a bond, a non-covalent bond. So the way that I think about shape is by using principal moments of inertia calculations. So what we do is we do molecular dynamics on the small molecule, and we generate a 3D conformational ensemble of the ligand. So thinking about all of the different shapes it could form in solution. We then use the program to find the longest vector through the molecule, and then form an XYZ coordinate. And then we divide I1 by I3 and I2 by I3, and then we plot this coordinate on a triangular plot, where each of the vertices represents a particular shape. So this vertice, for instance, represents rod-like ligands, or we could think about something like a baseball bat. This represents disc-like ligands, thinking about something like a frisbee. And then the other one represents sphere-like ligands, like a soccer ball. And these are just a few examples of what a rod-like ligand would look like. So this ligand is very elongated. This is an example of a disc-like ligand, so there's more symmetry in 2 dimensions. And then as you introduce things like bridges or stereocenters, you get more sphere-like character. So I did these calculations for all 1,600 ligands in the library and asked, okay, is there any shape-based recognition with this protein? So all of the ligands that didn't bind are shown in gray, and the ones that did are shown in red. And when I originally looked at this, I was not very hopeful that there was shape-based recognition that was occurring. But I started to dig a little bit deeper. And what I found is that there were actually 2 different shapes here that bound to this cysteine and Med25. And the first for this red group are these very linear-like small molecules. Then there's the second group shown in the green that we call a bent or a C shape. And then what was interesting was in the middle, this was actually our best lead ligand for Med25. And it has the ability to form both this bent or C shape, as well as a more linear-like shape. So there seem to be two shapes that bound out of many different options for this dynamic loop in Med25. So one of my next questions was, well, okay, how many of the molecules in this library have these shapes? So there are almost 300 ligands that had a linear-like shape, 7 of which actually bound to the target. There were 45 molecules that had this bent or the C shape, only 4 of which had any affinity for the target. And then there were 11 that had the ability to shape switch or form both a bent and linear shape, only 1 of which actually bound to the target. So we were really interested in understanding, okay, so it's not just shape. Shape is playing a role, but what are the other features that are defining this interaction with the protein? So this is work I've been doing with a really talented undergraduate. Her name is Chloe Markey. She's currently a senior in biomedical engineering. She's applying to graduate school this year. And she's really interested in computational work. So what she did is she took all the ligands that form this bent or this C shape, and she calculated over 300 molecular features. So things like molecular weight and hydrophobic surface area. She then did what we call feature selections to get a list of the features that correlate with the percent of binding to Med25. And she did this for all of the linear ligands, all of the bent or C-shaped ligands, and then for the entire library. And we used 3 different feature selection methods and looked for trends to make sure it was independent of the method that we were using. So what Chloe found is that there were actually 15 features for the bent or the C shape that correlated with binding. Most of those were related to hydrophobicity. For the linear shaped ligands, there are actually 14 features that correlated with binding. A lot of those related to the partial charge and the polarizability. And when we looked at the whole library, there were 21 features that correlated with binding, but there was no one particular dominant feature. And interestingly, there were 2 features that were common to all, one being negative partial charge and the other being polar volume. Polar volume, thinking about shape, I'm coming back to the idea that shape is important. I didn't describe this earlier, but Med25 actually is super positively charged. So we weren't surprised to see negative partial charges being a feature. Again, going back to electrostatics being important for the interaction. So coming back to my question, are there features that lead to this lock and key model that we can find in these dynamic proteins? And we did. We found that there were 2 ligand shapes that bind to this dynamic region in Med25, both of which have different features that correlate with binding. And this really led us to this framework for how to think about targeting dynamic structures. And it's the idea that there isn't just one lock and one key at any one place on a dynamic protein. We have multiple locks and multiple keys that work for those regions. So with that in mind, we wanted to see if we could push this idea just a little bit further and see if we can predict binding to this dynamic region using machine learning. So this is work that Chloe's been working on. So what she did is she took these bent or C-shaped small molecules and she divided them into 2 sets. One being a test set that was 25%, and the second being a training set for the machine learning model. She then took the training set. We used 4 different feature selections, 4 different machine learning algorithms for a total of 16 different models. We then selected any models that have an R² value greater than 0.7, And these were models that would be able to describe the tethering percentage or the binding percentage of the molecules. We then took the test set, which was not used in training the model, and asked, can we predict the binding of these 25% of the ligands? And for any models that can do that with an R-squared greater than 0.5, we call those models that can describe as well as predict the tethering or binding percentage. So she did this 100 times, and we were able to discover actually 55 models that were both describe as well as predict the binding percentage. And we chose 10 of those to see if we can predict new ligands that are even better than the ones we currently have for Med25. So the way Chloe is doing this is we're using the zinc database. So there's almost 5 million commercially available ligands in the zinc database. And Chloe wrote a code that describes what the molecular features are that lead to this bent or this C shape. So those features are having 2 rings, those rings being connected through an orthoconnectivity, and having an amine or carboxylic acid present to add the disulfide tail for the tethering reaction. So she filtered the zinc database for these features and found that there are around 50,000 commercially available ligands for us that we could purchase. She fed those into the 10 models that we built, and we now have new bent-shaped ligands that we can test that we predict to have even better affinity for Med25 in this dynamic region. So in addition to not only being able to know the features for these different shapes, we've been able to build a predictive machine learning model for the first time for a dynamic protein. So I've been telling you a lot about what we've been thinking about from the ligand side. So what about the protein? Does the protein actually form a lock of some sort for this interaction to occur? So we've been using a lot of molecular dynamic simulations to look at the structure of the protein when it's bound to the ligand. So for these bent-shaped or the C-shaped ligands, we only observed one pose or one binding mode in the simulations. And this is what it looked like. And I will tell you, when we got this back for the first time, we were all sitting around a table and we were like, how in the world is this molecule binding between these 2 dynamic loops? And when we built a surface model, what we observed is that there was indeed a pocket that was forming between these 2 loops. And it looks like a traditional lock, similar to what we saw with KRAS, a solvent-exposed pocket that this ligand is binding to. Going back to the different shapes of ligands that we had, one of our best ligands that we have for Med25 had the ability to form both the C or bent shape, as well as this linear-like shape. And we wanted to know, okay, which one of these shapes is actually biologically relevant? Which one is forming in solution? So using MD simulations, we found that there was one major pose for the bent or the C shape. And although this is a bent-shaped ligand, this pocket is different from the one that I showed before. And then there was a second pose for the linear-shaped ligand, where instead of binding in between these 2 loops of the protein, the ligand was forming an interaction with this alpha helix. So we wanted to know, okay, which one of these poses is actually forming in solution? In order to do that, we've been using NMR spectroscopy. So this is, what I'll be talking about today is proton nitrogen Hsqc. And this looks at the backbone of the protein. So we're looking at this nitrogen to hydrogen bond in the backbone. So we end up with an NMR spectrum for the apoprotein or the unbound protein, which I have shown here. This would be an example of a chemical shift that we would see in the NMR, and this would correspond to 1 amino acid. And then we would have a second one shown here in gray, And this would be a chemical shift that we would associate with a second amino acid. We would then repeat the NMR in the presence of the molecule and look to see if there were shifts for any of those chemicals or for any of the peaks. And this way allows us to interpret any structural changes that are occurring in the backbone. And we call these chemical shift perturbations. And we look at each amino acid in the presence and absence of the ligand to get structural insights. So this is my last question for you all today. So thinking about what we're actually reading here is this nitrogen to hydrogen bond. Which amino acid is invisible to us in this experiment?
Proline?
Yes, it is. So proline is a cyclic amino acid where there actually isn't a nitrogen to hydrogen bond present. So we can see all of the amino acids in the protein except for prolines using this experiment. So when we did this in the presence of our shape-switching ligand, what we observed is that for both of these loops, it wasn't that we had a chemical shift perturbation. Our signal was actually completely gone. And what that signifies is that the motion of the protein has changed for that amino acid. And that is consistent with ligand binding. We often call that broadening in the NMR. We not only saw that for the loop that the covalent bond is actually formed with, we also observed it for the secondary loop, which the ligand is forming interactions with. So we wanted to think about the other pose that we had, where potentially it was forming the linear-like shape and interacting with the alpha helix. And in particular, it was forming an interaction with this arginine 538. In our NMR spectrum, we did not see any chemical shift perturbations for this amino acid, suggesting that it was indeed this bent or this C shape that was forming in solution. So looking at this in a little bit more detail, we wanted to know what are the interactions that are forming between the molecule and the protein. So what this ligand is actually doing, and I think is really cool, is it's forming this bent or this C shape around the side chains of this valine residue. So it's basically hugging the side chains, which is shape-based recognition. In addition to that, what we see is that there's a methyl group on this molecule that's kind of pushing upon the second loop, forming what we would call a van der Waals interaction. And there is one potential hydrogen bond that could form. We didn't see it in the MD simulations, but it could form in between this carbonyl we found on the molecule and this arginine 509. So one of the ways that we can, in addition to using NMR to support our MD, we can also use synthesis to test derivatives of this molecule to support this binding mode. Some of you may be familiar with the term diversity-oriented synthesis. So we developed a diversity-oriented synthesis for this ligand. And what that means is that you want to start at a particular starting material, and you want to build your ligand through multiple steps. And then at the very last step, you want to introduce diversity. And this allows you to have a really efficient and really quick synthesis. So this is a synthetic scheme for making our shape-switching ligand. So we actually start out with 2 disulfides. This disulfide is symmetric, has carboxylic acids on both sides. We have a second disulfide. This has our tail, a dimethylamine. We do a disulfide exchange reaction where we generate this asymmetric tail, which has a carboxylic acid on one side and a dimethylamine on the second. We then do amide coupling reactions, which we were talking about right before, where we couple this carboxylic acid to a piperidine ring. One of the amines on this piperidine is Bach protected, that allows us to get selectivity between the primary and the secondary amine. So in the next step, we actually remove that Bach group or the protecting group using TFA. And then we do a second coupling reaction with the secondary amine and various carboxylic acids to make our final small molecule. And what we can do is we can alter this carboxylic acid at the very end to test different derivatives, all from the same starting material. So I want to give you a little bit of a sample of some of the information we've gained through these structure affinity relationships. And I mentioned before, when we were looking at the ligand binding pose, there was a methyl group that was kind of pushing up on that secondary dynamic loop. So we wanted to test, is that methyl group really forming that interaction? If we change it, do we see differences in the affinity? So we synthesize, these are several derivatives. We have our R stereoisomer, we have our S stereoisomer, so our different enantiomers. We also have a derivative that has no methyl group, as well as a derivative that has 2 methyl groups present. And similar to how we did the screen, we use what we call single point binding assays, where we have the same concentration of the protein, we add the same concentration of the compound, but we use 2 different concentrations of beta-mercaptoethanol. So we either use 1 millimolar or 100 micromolar, and that's our catalyst. And that's what allows the disulfide bond to form. So what's shown on the y-axis is the binding or the tethering percentage. And we have our different compounds shown on the right, both at 100 micromolar and 1 millimolar BME. And we want to see just a dose response, making sure it's actually the disulfide that's leading to the mass that we're seeing. So 22 and 22A, this was my original synthesis, and I was able to repeat, but for the parent molecule. So we have our R stereoisomer 22A. For 22B, the S stereoisomer, we saw a drop in the binding affinity. When we had no methyl group present in 22C, we were able to gain back some of that affinity. But when we had 2 methyl groups present, it was equivalent to having the S stereoisomer. Not only were these structure affinity relationships for binding to a really dynamic protein, but it also supported that the methyl group was important for binding and supporting our binding pose that we saw by MD simulations. So I'm going to summarize this work. So we're looking at targeting the cysteine residue in this dynamic loop. As you can see, it's moving in, it's moving out. There's a lot of motions in this protein. But we were able to observe features common to lock and key type protein systems, such as having shape-based recognition with the bent and linear ligands. We were able to discover additional features by using machine learning, such as hydrophobicity and partial charge. We can actually predict ligand binding using machine learning. We have examples of ligands that bind to this cysteine or bind to this region that have structure affinity relationships. And we know that between these 2 dynamic loops, there's at least 2 unique pockets for ligands, those that bind to the bent shape ligands and those that bind to the shape switching ligands. And that really comes back again to that framework of, well, how do we target these dynamic structures? Dynamic proteins are ligandable. They're not inaccessible to us if we think about using covalent ligands. At any given binding location, think about there being more than one lock and more than one key. There's more than one possibility for these proteins. Also thinking about each of these locks, each of these keys has its own features that are important for binding. Instead of trying to put them all together, let's separate them into their own entities to understand more about their features. And I think one of the really important things to take away too is thinking outside the box can really help us solve a lot of scientific problems that have eluded scientists for decades. And all of this was possible because of the formation of a covalent bond. So what I'm going to be working on at Notre Dame is using this framework and all of these ideas that we've discovered to hopefully discover new proteins that are in these 60 to 80% that we can make small molecules for, those that are really important in cancer metastasis, for example. So I'll stop there as far as research. And this is my postdoc lab at the University of Michigan. My advisor, her name is Anna Mapp. She's really wonderful. And these are some of the great individuals that I've been able to work with in my postdoc. We have a lot of really great collaborators, as well as a lot of funding sources. And I could stop here if people have research questions. I also have a couple slides about my journey as a scientist too.
Oh, yeah, I was saying you could go ahead and show those and we could take questions after.
Okay, do that. I'm now stuck in the chat.
Are you stuck?
Yep, I'm good now.
Oh.
So I wanted to tell you all a little bit about how I became a scientist, because it was not ever on my radar to be here today. So I actually grew up in a really small town in rural Kentucky. There was less than 5,000 people in the town I grew up in. My mom was a stay-at-home mom. My dad worked in a factory. Both of my parents were high school graduates. I grew up on a farm. I loved petting the various animals. Here's a miniature horse. I was always playing with the kitty cats. I was bottle feeding the calves, just growing up living in life on a farm. And I, from there, of course, finished high school, but I really had no idea what I wanted to do. And there wasn't a lot of job opportunities in my hometown. I was in the band. I did a co-op in high school to learn about pharmacy. It wasn't really my thing. It just wasn't exactly what I wanted to do. But I had this really phenomenal chemistry teacher. Her name was Miss Hall. And she helped me fill out my one college application to Western Kentucky University. I stayed after school one day, she helped me fill it out, and she made a phone call to make sure I got into a research program at that school. And I started doing research with this wonderful individual. His name is Professor Kevin Williams. I had no idea what research was. I honestly had no idea that you could even major in science in college at that point. He took me in as a freshman and guided me really throughout my college career. I was a major in biochemistry. I completed the research program my chemistry teacher got me involved in. I was able to travel for the first time. I had never been on a plane. He was able to give me a flight out to California to present my research at an ACS conference. I remember sitting in his office being like, how do I get on a plane? Like, how do I pack? It's my first time really traveling. I worked part-time through college as a pharmacy tech from my co-op experience in high school, but I was also able to work part-time as a research assistant in his lab. And he was just amazing in guiding me, and I loved doing research. I loved learning, and I never wanted to stop. And that's what really brought me to graduate school. So I applied to several, I think it was 7 or 8 different schools. I got into 4 of them, and I ended up going to Duke University. There, I had an absolutely wonderful, again, mentor. That'll be a common theme throughout my story. Her name is Amanda Hargrove. And this is a picture of me and her after my master's exam, a picture of me after I completed my PhD. One of the things I loved about graduate school is that I got to do a lot of outreach. So I love making liquid nitrogen ice cream. But I also got to take on a leadership role for the first time. And this was a speech I was giving at the dean's office about the importance of mentorship and how important it had been to me throughout my life. And I got to travel even more. And these are just some of the pictures from the various, it's one of my favorite parts of science is getting to go places, see new things, and talking to people about science. And it wouldn't have been possible without me going to college and really having this career. And I've also made a lot of really wonderful friends across the United States from that experience. So I was still just learning at this point. It was really at the end of graduate school for the first time I knew what I wanted to do. And I wanted to be a professor. It had all of these wonderful individuals who had helped me so much, and it wouldn't have been possible without them. So I was 28 when I knew what I wanted to do for the rest of my life, and that's perfectly okay.
That's right.
So why did I want to be a professor? I love teaching. I wanted to have my own research lab. In my own lab, I can study whatever I want. And I also loved, again, the traveling aspect and just really getting to see the world and meeting all of these wonderful people. So for most positions to become a professor, you have to do a second job, which is called a postdoc, which is what I'm doing right now. It's pretty similar to an apprenticeship. Instead of working kind of under your advisor, having a boss, you work more like side by side with them. So that's what I've been doing here at the University of Michigan. I've been learning a lot about biology, biophysics, biochemistry. And that's really how I got here. It wasn't always my destiny. I started really as a farm girl in small town Kentucky, graduated from high school, got my bachelor's degree from the university 30 minutes down the road from where I grew up, got my doctorate at Duke, finishing my postdoc here at the University of Michigan, and soon to be a professor at the University of Notre Dame, which is just crazy to me to even think that was not a possibility for me when I was growing up. So a lot of people ask me, what do I wish I would have known then that I do now? And there's really two things. One is I have a lot of courage. I was a lot of first, and I wasn't even thinking about it when I was going through it, but I'm a first generation college student. I was the first woman in my family to have a career. I was actually the first family member to even leave my small town in Kentucky.
Wow.
I was the first PhD student for my advisor. So many firsts, and I wasn't thinking about it. I was just following what I wanted to do at the time. So in addition to having courage, it's really relying on mentors, sponsors, and coaches. All of the mentors, these are just some of the pictures of them. They have always sought out phenomenal opportunities for me. And leaning on them and asking them for help and going to them and saying things like, how do I get on a plane? But having those people to give you feedback and help you find and get to your next step is so important. Their encouragement is just priceless to me. And these are just a few of those people that have got me here today. And this is my last slide. I always, being that mentors are really important to me, being a mentor to other people is also really important to me. And these are just a few of the undergraduates that I've mentored throughout my career. And they're doing a lot of different things. They all did research as an undergraduate. Some have gone on to medical school, some have gone on to graduate school. Bilva is now a software engineer at Amazon, making a lot more money than I will be making. But research isn't just important for being a professor. There's a ton of different careers, from engineering to physical therapy, to forensics, to even science communication. So if you're thinking about graduate school, it's not just being a professor. There's a lot of different things that you can do with a PhD. Mm-hmm. So I will stop there and say thank you. My email is here. I'm more than happy to talk to anyone about graduate school, more about targeting proteins. I also have a Twitter. These are my two wonderful dogs. And these are, again, some of the wonderful people that I've had in my life.
Wow.
And, yeah.
Wow. I love, love, love your story.
Thank you.
That is really cool. Wow. That's really inspiring. I hope some of our students and others are inspired by your narrative, your story. It's really, really something to think about, to admire. We would like to open it up. So anyone could ask questions either about Brittany's, Professor Morgan's professional journey, life journey really, and or her research. So you could just unmute yourself and ask questions. As people are getting their questions together, I have several. First of all, I think we definitely need to collaborate on stuff. So you and I will be talking some more for sure. But in terms of the research, several questions. One on since it came up in our prior conversation before the seminar began, the method that you use to make the amides, it's a well-known method and stuff, but I think one of the disadvantages, even though it's successful, one of the disadvantages, isn't it that it creates a lot of byproducts that you have to then try to, purify extensively by chromatography?
It is a pain.
Yes, that's what I remember.
So not only do we do an extraction afterwards, we typically have to run at least 2 columns to get it pure enough. Yeah.
Yeah, so that's one of the things I told my class when we set out to investigate, at least 4 different methods for making amides, is that the reason why we try to develop synthetic methods is that we want to make things more efficient, and we want to have more tools, a methodology. A method is a tool. And so, if we could find a better method that's easier, less work, no need for chromatography, that's the sort of thing that synthetic methodologists, I guess, would want to go after. So that's one point I wanted to bring out.
Absolutely. It takes, in total, it probably takes us close to like 15 hours to purify.
Yeah. So anybody else has questions, comments, either on the science or the life journey? I have other questions in the meantime, but I want to make sure other people do get a chance.
I'll ask a quick question here.
Sure.
Thank you for sharing your story and the information. I'm from a small town in Michigan and kind of had a lot of similar experiences as well. First time in my family to be a college student at a 4-year university.
Wow.
To make it. So you guys can make it. You just gotta.
That's right.
Be persistent and ask questions. You don't care how dumb they are. You just do it. So thank you for sharing that.
Mm-hmm.
So you said that there's some proteins that are not ligandable or druggable. I wonder if you could say a little bit about why that is. Maybe you could share what is it? Why can't those be targeted in some way?
So for the past, how many ever years we've been medicinal chemists, probably since the 50s, 60s, people don't want to use covalent ligands, and they have a lot of potential toxicity problems. And that was it came out from the story of aspirin. Aspirin leads to several byproducts that can be toxic. And people, medicinal chemists, up until really the past 5 years, refused to make covalent ligands and put them in libraries that we use to screen proteins.
Mm-hmm.
And that's how we got here, where we have 60-80% of these really dynamic proteins that we've been screening with non-covalent ligands for many, many years, and we're not able to find leads. And we're just now really appreciating that covalent ligands can work. We have FDA-approved drugs for KRAS, as well as several other targets now. I think it was last year, 40% of the small molecules FDA-approved were covalent. And so it is a promising strategy, but if you were to go buy a library of covalent ligands, there's not that many that we can go purchase. So that's really the problem that we're at right now, is we don't have them commercially available, but we know that they have a lot of promise for these undruggable and unligandable proteins.
But there's still some that are out of reach, though. I guess I'm asking about what makes them out of reach. They just don't fit the traditional models of how we're trying to attach things? Or what can you say about this unreachable group? Anything about them?
I would estimate around 50% of the unreachable group we can reach with covalent ligands. There's still going to be a subset of those that don't form enough structure for a small molecule to be able to target it. And that's one of the things I really want to explore in my independent lab, is really pushing that boundary. How many of these can we target? How unstructured can these regions be? And that'll really give us an idea about those sets of proteins.
Yeah, I think I read recently, there was something in either CNE News or someplace about the resurgence of covalent drugs. So I'm pretty sure you're probably familiar with that literature.
Yes.
Yeah. And so one of the things I thought about while you were talking about the lock and key is, are you familiar with, there was a controversy, if you will, mini controversy that came out of a study done at Harvard that made us sort of reconsider the lock and key model, and that basically that water could gum up the works in terms of the lock and key model. And so I'm wondering if in your modeling of these ligands, are you taking that into consideration as well?
When we do molecular dynamic simulations, they are in the presence of a water box.
Okay.
But that is a really important aspect, is for these ligands to bind, whether it's in a pocket or even a surface exposed pocket, those water molecules do have to be displaced, and that takes energy.
Yep, that was the finding. Yeah, yeah, that was, it generated some, debate between George Whitesides and Jean-Marie Len.
Yep.
Yeah, so, okay. So you're familiar with that in your modeling and calculations and stuff?
Yes.
Yeah, okay. Other questions? So, I think this is more of a comment than a question. I think we've actually had two of your professors, or at least collaborators, Anna Mapp, I believe some years ago, was a speaker here. And I believe also Heather Carlson. She's, yeah, I think both of them were actually speakers some time ago. Yeah.
Wonderful women who have been my mentors.
Yes. All right. So you'll be here in July?
I will be. And if people are interested in graduate school at Notre Dame, I of course have a slide for you all.
Okay, good.
If people are interested, feel free to reach out to me. The next applications will be due at next December or next January. So we've already finished admissions for this cycle. But we have a chemistry and a biochemistry program. We also have a biophysics graduate program and an integrated biomedical sciences program. I'm happy to talk to anybody more about that.
Yeah, one of my students right now is in biophysics. She's in my organic chemistry class. Maybe she might be interested. Yeah. So have you, I'm assuming you started, even though you're not physically here, you started, they've given you a place, you started looking for students to join the group. Is that sort of thing going on now?
Yes, I actually have the experience of building my lab from scratch. It's 4 concrete walls. So I've been working with designers. It's been a really fun process.
From scratch?
Yes.
Wow.
It's been fun. But yeah, we're looking for students, anybody who's taking a gap year next year, potentially. I'm looking for people to do synthetic chemistry in my lab.
Oh, I know at least one student doing a gap year.
Yes, have them reach out to me.
Okay,
And
I will.
I have my first grad student already.
Okay.
Speaking of first, I am someone who does take a lot of risk.
Yes.
So I do have a grad student, but yeah, we're looking to build the group and doing similar work to what I described today.
All right. Well, sounds good. It's great listening to your research. It's wonderful hearing your life story. It's really inspiring. And we will definitely be in touch.
Thank you all so much for having me. It was really great.
All right. Have a good afternoon, evening.
You too.
Take care. All right. Bye-bye.
Hey, Desmond.
Yes, sir.
We should have every speaker just like have a slide or two on their life story. That would
On
be
your life
really
story. I know, right?
We got that from Hannah.
Yes.
Just a really cool thing to.
A human interest story. Yes,
Yep.
Absolutely. Yep. It was really inspirational, man.
Yeah, just to hear it, right? Because
yeah,
people just might put faculty and all you guys, whatever.
Yes.
I don't know what they're thinking.
Right.
But they're real. Yeah.
First time she was going on a plane, she had to ask. I mean, it's funny and it's really cool.
Absolutely. That was, and I think that's what we're trying to do with this program, right? Is just make it real. Make science real, chemistry real. There's real problems.
Exactly.
And find that frontier of ignorance.
Right. That's right.
And create opportunities. So thanks for doing this.
Yep. Yeah, it's also interesting that she's, I can't remember now how I reached out to her, whether she was already on the Notre Dame site or did I see her at the University of Michigan site? I can't remember for sure right now, but yeah. She's in that.
Appreciate what you're doing and appreciate that she did it. That was a great presentation. I thought it was very, she went at a good speed and good graphics and had a nice flow to it. Yeah.
Oh, yes. All right, man. Thank you. Have a good evening. Stay warm.
Very important.
Yes. Take care.
Bye.
Bye.